Alright, in this video we're going to talk about cells. This is going to focus on the macromolecules that make up the different parts of the cell, the enzymes within the cell and like what they do, the types of cells you will find, the important organelles, how things move through the cell in terms of across the membrane, and then finally we're going to finish by talking about how energy is made and the important reactions of photosynthesis and respiration. Alright, to start with, we're going to talk about kind of the different levels that you can come across. So everything starts out as an atom, okay, and those atoms together make molecules. The molecules then create, along with the atoms, what we call our macromolecules. These are our polymers, so your carbohydrates, your proteins, your lipids, and your nucleic acids. We'll talk more about those in a second. These macromolecules or polymers create your organelles, which make your cells. Okay, then from there they can become tissues, they become organs, organ systems, organisms, and then you can go bigger: species, population, community, ecosystem, biome, and biosphere. But for this segment, we're just going to focus on cells and what makes up cells and the parts in there. So first of all, we're going to talk about the four macromolecules. Okay, the first one we're going to talk about is a protein. It's made up of an amino acid. So a couple important terms. First of all, monomer is what we use to describe what makes them up. They're like individual building blocks. The polymer is going to be the macromolecules. These are the things that are created from the monomers coming together. So amino acids are the monomers of proteins. Now, the other thing that's important about this is what do they do? Enzymes are types of proteins which help with chemical reactions. Hormones are like signaling proteins. Proteins can have a structural job. Basically anything in the cell that needs to be done can be done by proteins. The second macromolecule we're going to talk about is carbohydrates. They're made up of something known as monosaccharides. As fancy as this sounds, it means just a simple sugar. So an example of that is glucose. These are your short-term energy sources. So this gives you a burst of energy, it's quickly burned, and then moves into the lipids. Okay, lipids are made up of fatty acids, and these are your long-term energy storage. Okay, so a lot of times, like if someone's trying to lose weight, the harder part in losing weight is going to be the fat, because it is seen as a long-term energy source. And the final final macromolecule is nucleic acid. It's made up of nucleotides. This one kind of makes sense because they both start similar. Nucleo and nucleic, they sound similar. Their most important job is going to be storing genetic information, such as with DNA. They also help with protein synthesis, but their main job is going to be with storing the genetic information. Okay, so here's images of each one of these. The top one up here is your protein. The second one is your carbohydrate, or your sugars. The third one, actually no, I take that back. That second one is not your carbohydrates. That is your nucleic acid. So let's try this again. The first one is your protein. The second one is your nucleic acid. Okay, the third one is your carbohydrate. And your bottom one down here is your lipid. Okay, these are just examples of the macromolecules made up from their monomers, and kind of just showing you an illustration of it. Alright, so I mentioned earlier enzymes. Enzymes are specialized proteins that run reactions, okay? They are known as catalysts or biological catalysts, okay? A catalyst is something that will reduce the activation energy and make the reaction run faster. Alright, there's some important key parts. You've got your substrate, which fits into the active site of the enzyme. So in this case, here's your substrate. This green shape thing is going to fit into the active site here. We call this the lock and key model, which basically means that one key fits in each lock. So there's one substrate basically for each enzyme. Biochemical reaction will take place, and each substrate is very specific to its enzyme. Now if we go back a slide, if you notice down here in this little GIF that's going on, you'll notice that we're lowering the pH of this particular thing, and that the the enzyme is basically changing shape. We say that it's denaturing, which means it's basically being destroyed. So denature equals destroy or destroyed. And if you change the pH or temperature or both of an enzyme, it will denature it. Basically means it won't work anymore. So enzymes work in a particular range of temperature or pH. Alright, moving on to cells now. We have two general types of cells. You have your prokaryotic cell and your eukaryotic cell. Prokaryotic cell, the biggest thing is that they have no nucleus. They don't have membrane-bound organelles at all, but the big thing is they don't have any nucleus. This is going to be your bacteria. Eukaryotic cells do have a nucleus. Okay, they also have the membrane-bound organelles, like your ER, your Golgi body, a lot of those other things, and this is everything else. So they're more complex. This is your fungi, your plant, your animal, things like that. Those are your two general broad categories. Now, for the relationship between them, we have something known as the endosymbiotic theory, and this basically tries to explain how eukaryotic cells came from prokaryotic cells. So the idea in this is that at some point you had a prokaryotic cell that became eukaryotic. Okay, so this is a cell that would eventually become eukaryotic, and the way it became eukaryotic is it swallowed up other prokaryotic cells. That eventually became your mitochondria and or your chloroplast. Okay, so those two organelles are kind of ways that we look at evidence for this theory, because these have their own DNA, they have their own ribosomes, they can make their own proteins to an effect. But the idea is that this, whatever this, this primitive prokaryotic cell is here that became your eukaryotic cell, swallowed up these other prokaryotic cells and kind of made them essentially organelles. So that's the endosymbiotic theory. Again, it just explains the relationship between prokaryotic and eukaryotic, and how eukaryotic are more complex. Alright, so then eukaryotic cells can be divided into plants and animals, and there are some unique features to both. Okay, so in an animal cell, you have centrioles that are only unique to animal cells, and you also have lysosomes. These are also unique to animal cells. Plant cells have two also. They have this chloroplast and the cell wall. Okay, they have also, you may read, they have what we call a central vacuole, which is what I started here. It's not really unique except for the fact that it's very large. Animal cells do have vacuoles, but they're not nearly as big as the central vacuole. So really the two unique things to plant cells are chloroplast and cell wall, and for animals it's centrioles and lysosomes. Okay, so now let's look at the organelles. There seems to be a lot of them, but let's go over these briefly and try to think of bite-sized things to help you remember them. So the nucleus, that's your control center. It stores the DNA, helps dictate or control other parts of the cell. The nucleolus makes the ribosomes. It really has no relationship in terms of with the nucleus. A lot of people think that the nucleolus is where the DNA is stored. It's not. The nucleolus's job is the ribosome. Ribosomes are for protein synthesis. They make our proteins. Endoplasmic reticulum, or ER, is basically pathways for our lipids and our proteins that are made there. The Golgi body is going to be like your packaging center. It takes in, modifies, and repackages all the proteins and lipids and sends them out, typically outside the cell. Lysosomes are the trash cans. They take in the broken parts, they take in the foreign objects that need to be destroyed, and they break them down. The vacuoles are like your book bag or your bedroom closet. They store stuff. They store waste, they store food, all sorts of stuff. Now, like I mentioned, animals have smaller versions of it, whereas plants have one large one. The cell membrane is very important. It basically holds the cell together, and it controls what goes in and out. the mitochondria. This one you definitely should know. It's the powerhouse of the cell. It's where cellular respiration takes place, and it makes ATP, which is our currency. The chloroplast is where photosynthesis takes place, makes sugar for the plant. It's only found in plants. The cell wall, again, also only found in plants, adds an outside border, like boundary almost, and gives it support. And finally, the centriole, it helps in reproduction through the process of mitosis. And again, it's only found in animal cells. The lysosome, I didn't mention earlier, is also only found in lysosomes, or in animal cells, excuse me. There's a lot here, but if you can remember those bite-sized little bits about each organelle, then you should be okay. Alright, moving on to the cell membrane. This is a very important part of the cell because it does control what comes in and out. And the term we use for that is what we call selectively permeable. And this basically means that only certain molecules can come into the cell or leave the cell. And it comes down to the way that it's arranged. So let's look at the different parts of the cell membrane. So the main core part of the cell membrane is made up of a bilayer, 2 layers, of what we call a phospholipid. Okay, this looks like looks something a little bit like this. This is a crude drawing, but you have the head that is polar. Okay, so this is the parts that are on the outside here, where I've drawn the lines. They like water. They're hydrophilic. Okay, so that means that they're going to interact with the outside and the inside of the cell. They're going to form the borders of the bilayer. The bottom part, or the tail, is nonpolar. Okay, this is where I've started here in the middle. Okay, so you create this nonpolar environment inside the bilayer that helps separate the inside from the outside. Okay, the other things that we have in there are some proteins to help move things in and out of the cell, and also to help with signaling, communication. And one thing that's not mentioned in there, but that is important, is cholesterol. Okay, cholesterol helps with the fluidity, like how well it moves, how well it flows. Too much cholesterol, it gets really rigid. Too little, it gets too fluid, moves around too much. So those are the key parts. The phospholipids themselves really help define what comes in and out. Alright, so now we're going to talk about how things actually move. Alright, the first thing we're going to talk about is diffusion, because this is kind of the general idea of movement. Alright, diffusion is a type of passive transport. We'll talk more about that in a few minutes, but passive transport moves from high concentration to low concentration. You'll often hear high to low, okay? And what happened here is at the beginning, if you were paying attention, you had a bunch of molecules down here, and over time they spread up and out. So you're moving from a very concentrated, very high amount of particles, and they eventually spread out to where there's less. Okay, that's the idea of diffusion. It spreads out. And so passive transport, like I mentioned, diffusion is one type. The other type is what we call facilitated diffusion, which is basically the diffusion with proteins. So it requires something that can't go through the membrane itself, but has to have a protein instead. Right? And then active transport is something that requires energy. Okay, so active transport requires energy, whereas passive transport does not require energy. Okay, so let's look a little bit more at those in depth. Okay, so like I mentioned, we move from high to low. We also say we move with the concentration gradient. With the concentration gradient means that you are moving from high to low. You don't have to have energy. Okay, like I said. So we're moving with the concentration gradient. We're going from high amounts to low amounts. Examples we talked about before are diffusion, facilitated diffusion, also osmosis, which is one we'll talk about in a few minutes. Osmosis, that's basically diffusion of water. So they're just different ways of describing diffusion or more specialized cases. And then active transport is opposite. It moves from low concentration to high concentration. So it's like going into a crowd. You're going against the grain, and this is definitely going to require energy. Alright, what you should see up here, one example is what we call a protein pump. This is similar to facilitated diffusion, except for obviously it's active. and you're moving molecules from a low concentration to a high concentration. This is what this picture is showing there. And then if you have to move large molecules, you have two ways you can do it. You can do endocytosis, where endo means basically enter, you're bringing things into the cell, and exocytosis, which is where we're moving things out. So exocytosis, or exit, okay? And they both require energy. That's what the bottom picture here is showing endocytosis. And there's two types of endocytosis. They're not very important, but the terms are good to know. Phagocytosis deals with solids, and pinocytosis deals with liquids. So these are types of endocytosis. It just depends on what you're bringing in. Alright, like I mentioned before, osmosis. We're going to talk about the different conditions you can get with osmosis, but osmosis basically is diffusion of water. Okay, so it is passive. This is, like I mentioned before, it is a passive, this is passive transport, and you can see three different situations with osmosis. You can see what we call hypotonic. Okay, hypotonic sounds like hippo, so it's going to get big and it's going to burst. And the reason why this happens is that you have less water inside and more water outside. So remember, it moves from high to low in terms of water movement, so the water is going to move in. Isotonic, you have equal amounts. So this one, you'll have equal amounts inside and outside, so the water nothing will happen to the cell. It will move like it needs to. And then hypertonic, this is like a sugar crash. Like you come off a sugar crash, you just feel deflated, you feel out of energy. And that's really what happens here. It shrivels up. So you have more water inside the cell and less water outside. So again, it moves from high to low, and all the water will move out. Whereas with hypotonic, it moves in. Okay, same thing will happen with plant cells, but because of the cell wall, the cells won't necessarily change as drastic of shape. The cell wall gives it some form still. Alright, the other way we can look at this is by solute. And one thing to keep in mind is that if you have high amounts of solute, you have low amounts of water. They're basically inverse of each other. And the opposite is true. If you have low amounts of solute, you have high amounts of water. Okay, so we can actually look at these different hypotonic, hypertonic situations from the point of view of the solute. So with hypertonic, you have more solute outside. Okay, so that means less water. Okay, remember we talked about the more solute you have, the less water you have, so that's more water outside, which is, or less, excuse me, less water outside. That's going to cause it to shrink because all the water will come from outside to inside. Hypotonic means you have more salt inside, so you have less water inside, which will cause it to swell. Okay, so wherever you have the more solute is where the water is going to move, basically. So you need to be able to understand the point of view from where the water is coming from, but also if they describe the solute, it's a good point of view to understand that. And like, I didn't mention iso because iso is the same either way. Alright, so we're going to finish this up by talking about two very important reactions. You have photosynthesis, which is basically this plant taking sunlight and turning it into glucose, or sugar, which is their energy, or their food source, excuse me. And then cellular respiration, where you take that glucose and you break it down to make energy. Okay, and one thing that's really important, we'll look at the equations here in a minute, is that the products of photosynthesis become the reactants for respiration, and vice versa. Okay, so it's almost like a cycle in a way. So the products of one are the reactants for the other, and so they are very, very interrelated. And one thing I want to point out is plants actually do both. Animals just do respiration, but plants also do respiration because they need their energy. Okay, so here are the equations. It's important to understand how to identify them. So my biggest suggestion is to look for the sugar. So if you look in photosynthesis, your sugar is going to be a product. It's what's made, because the whole point of respiration is, or excuse me, the whole point of photosynthesis is to make sugar. So it's going to be on the right side of the reaction as a product. Whereas for respiration, it's a reactant. We're using that to we're going to break it down, and so it's going to be at the beginning. It's going to be on the left side of the reaction. So based on where the sugar is, that should tell you what type of reaction it is, whether it's photosynthesis or respiration. Alright, so this finishes up this section on cells.